So that's what the patterns you should see under drift. The obvious questions is, okay, what patterns should you see under selection, right? There's all sorts of really interesting things come up. Again, I point out that if you're interested in looking at some of this stuff in detail, it's in Chapter 8. Here's the printouts of some of those chapters. You're welcome to grab that. And like I said, if you want to take that home tonight and just return tomorrow, that's fine as well. So here is the overview. I want to talk about types of sweeps. Remember I mentioned this coalescent? The coalescent simply says, if I have neutrality and a constant population size, what do I expect those genealogies to look like? If I then throw mutations on those genealogies, that generates the site frequency spectrum and things like that. What's the impact of selection on that coalescent structure? I want to talk about hard versus soft sweeps. I'll say a little bit about the pop gen of sweeps. Much more of the details are in Chapter 8. Then I want to talk about standing sweeps and recurrent mutations. That is, a pattern that's obvious is if you look at a lot of genomes, in regions of reduced recombination, there's less variation. There are two models that account for that. Number one, recurrent sweeps. That means lots of adaptations going on. Cool. Number two, background selection. New mutations arise are deleterious, they're removed. Both those generate essentially indistinguishable patterns. Wolfgang Stefan, a really bright guy at Munich, spent 20 years trying to distinguish these, and he still says, we're not quite sure. So we'll talk about those. We'll talk about the impact of recurrent sweeps. A cute example is codon usage bias in Drosophila. We had this notion of when is an allele effectively neutral? When for any absolute value of S is less than 1. The noise that's generated by drift is larger than the deterministic signal from the strength of selection. So that allele basically behaves as if it's neutral. The really cool thing is in Drosophila, codon usage bias is sitting right around for any S equals 1. So I have a local reduction in the effective population size. All of a sudden, I get neutrally generated codon bias. If my effective population size stays the same, I get selectively generated codon bias. So we see some fun signals in that, and that leads us into something called the Hill-Robertson effect, Bill Rill and Alan Robertson, and this really is a critical thing. To frame the Hill-Robertson effect the following way, I think the neutral theory was developed by Moto Kimura mainly and other people. Tomoka Oda did a huge amount of work on that as well, and her work is really important. And it was basically said that if you look at standing variation and differences between species, the vast majority of that is due to neutral genes. So if I look in you and I, we differ at 20 million sites, segregating sites. Kimura would say the vast majority of those are neutral. Oda would say not quite true. Some of those are slightly deleterious, but nonetheless, they're not advantageous. If I look at a DNA sequence between, let's say, us and a salmon, and I see, 500 substitutions total, Kimura would say the vast majority of those are neutral. Certainly, there are adaptive substitutions, and some are important, but the vast majority are neutral. That's the standard neutral theory. The new version of the neutral theory is the neutral theory works because there's so much selection. That sounds counterintuitive. The neutral theory is built on selection, but selection removing deleterious alleles. The new version, based largely on Hill-Robertson, if I scan across a genome, most sites in the genome are near sites which have been selected. Maybe it's for advantageous, maybe it's to remove deleterious mutations, but the net result of selection at nearby linked sites is to lower the effective population size. What happens when you lower the effective population size? A larger category of selection coefficients now become effectively neutral. So one reason we see the neutral theory largely holding is not, in my opinion, because of the absence of selection, but rather because selection is so common, which is really counterintuitive. And a point we'll come back to. And again, a lot of the details in here, if you want some of the more fine details, again, they're all given in Chapter 8. Chapter 8, I think, is one of the more dense chapters in the book. That and the chapter on mutation, selection, equilibrium for quantitative traits, I think, are the two heaviest ones. So when an allele is linked to a site under selection, its dynamics are altered considerably relative to drift. A neutral mutation can hitchhike to high frequencies. A classic paper is Maynard Smith and Haig, but actually the term hitchhiking was used in a paper before theirs by Schaefer. But this notion of hitchhiking and the notion of sweeps are connected. So a sweep refers to the consequence of being on a chromosome near a site where an advantageous mutation has been fixed. The net result of the sweep, as we'll talk about, it's called the sweep because you swept away all the variation. So one classic signal is a dip in the amount of variation as you scan across the chromosome. A partial sweep occurs when an allele is increasing under selection, but it hasn't gone to fixation. It either is in the process of going to fixation, or it's overdominant, so it's being changed from a low frequency to some equilibrium frequency, or selection favors it and the environment changes. That rapid change relative to drift in allele frequencies over some small interval is called the partial sweep, and that still has dramatic consequences on the way the genome is structured, as we'll talk about. A polygenic sweep occurs when adaptation occurs not by a few alleles going to fixation, but rather by a large number of alleles all having small shifts in their allele frequency. A polygenic sweep doesn't leave a really obvious signal, and a big debate that's still kind of going on are signals of so-called hard sweeps are unusual in humans, and people have argued that maybe most of the selection in humans is due to polygenic sweeps, which don't leave the classic signals we'll talk about. They leave other signals we'll talk about using other methods. So a key here is favorable selection can operate in many different ways, and those different ways leave different signals and therefore require different tests to detect them. Key point number one about tests of selection is rejecting a test of neutrality doesn't mean selection. It very often means you have rejected the equilibrium assumption. Key part number two, different types of selection require different types of tests. There's no one omnibus test that detects all selection. So you could do 5 or 6 different tests on your data. One may give a big signal, the others may all fail. That could be simply sampling noise, but we'll talk about which tests we expect to be important for detecting hard sweeps, soft sweeps, ongoing sweeps, recently completed sweeps, polygenic sweeps. Those are all different signals. And one of the reasons there's so many tests is there's no one jackknife test that does all. So let's then talk, first of all, about what a sweep does on the pattern of ancestry, that coalescent structure. So a key concept is TMRCA, time to most recent common ancestor. And it's something that means if I take a sample, how long do I have to go back to a common ancestor? If that sample is generated by drift, I have a certain time to go back because it takes 4 N generations to do that. If that sample is generated by selection, that time may be much shorter because if I have a site linked to a favorable site, that favorable site sweeps through the population very quickly, pulls sequences along with it, and the time back to the common ancestor is much shorter than I expect relative to drift. Shorter time means less variation, longer blocks of LD, as we'll talk about. So yeah, shorter time to most recent common ancestor, less variation. So think about it. If everybody here has a common ancestor 1 million years ago, I take that pattern, I throw mutations down on it, I have lots of variation. If everyone here has a common ancestor 2 generations ago, I have almost no variation because there's very little time for mutation to arise. So as I shrink the time to most recent common ancestry, I shrink the expected divergence. Also, If I shorten time, what does that mean? It means less time for recombination to break up blocks of linkage disequilibrium. So time allows mutations to accumulate. It allows LD blocks to shrink. Good example. You take Europeans and Africans. Africans have higher rates of nucleotide diversity and have shorter blocks of LD. Why? Deeper time back to a common ancestry. Europeans are a subset on one of the migrations out of Africa. They have a shorter time back to common ancestry. Shorter time means, number one, less time for mutations to accumulate relative to Africans, so less variation. Number two, less time for recombination to act, therefore longer LD blocks. In Africans, there's been more time back to common ancestry, so more time for recombination to shave those LD blocks down. So time to common ancestry sets a lot of features in molecular characters. Now, directional selection gives me a shorter time back to common ancestry. Long-term balancing selection gives me a longer time. The population balancing selection means selection is actively maintaining two alleles. They're around longer than I expect under drift. Therefore, there's a longer time back to common ancestry. Therefore, they should show more variation. So here's the basic idea. If I have a kind of a sliding window, and let's say I'm measuring nucleotide diversity, it's kind of a general measure of variation. So I've got a sliding window. Any single site will be very noisy, so I've got a sliding window. And what I see is I see a big dip. That's a classic signal that you expect from a site under directional selection. Likewise, if I see this, that's a classic signal you expect for site under balancing selection. However, if you see either one of these by itself, that's not sufficient. Why? That could be a mutational cold spot. That could be a mutational hot spot. So the way you control for that is you have an outgroup. Take maize and take teosinte, for example. So these patterns are very suggestive of that. Now, there's a problem with balancing selection. Balancing selection, if you keep alleles around longer, what happens? You've got more recombination, and therefore the region of the signal shrinks and becomes smaller and smaller and smaller over time. So this signal here is actually rather fleeting because the window that's been kept around, has a long common ancestry, gets smaller and smaller and smaller as recombination breaks it up, and therefore you get less and less a signal. So it's actually harder to pick out long-term balancing selection. But here's an example. Here's the fast-slow alleles in alcohol dehydrogenase. Right there at that arrow, that's a nucleotide substitution that gives you an amino acid change that gives you two different alleles. They're called fast and slow because one moves faster on a starch gel than the other. So in the old days, the fast and slow alleles were fairly well known. So you get a big spike there of increased variation, and that's suggestive of balancing selection. That's not the whole story, because you can basically show that all that excess variation is not between alleles, but it's within one allele. So it's not a complete story yet. Something is going on there, but this is the signal you expect from balancing selection. A classic example of a sweep is maize and its ancestor teosinte. I'll show you a slide of teosinte a bit later on. What you can then do is basically look at a gene called TB1. Teosinte branched 1 is a mutation in maize that takes that single stalk maize and makes it a multi-stalk plant. Teosinte was a multi-stalk plant. It's the ancestor to maize. And so this is kind of a back mutation. And so people thought that TB1 is a gene involved in domestication. It takes this multi-branch plant architecture and makes a single branch. So then if you scan around teosinte branch 1 with the nucleotide diversity, you see a couple things. So number one, the dashed line here is maize. Number two, the solid line is teosinte. Why over here is maize generally less than teosinte? Went through a founding bottleneck, right? I got this big teosinte population. I pull out a sample to make modern maize. That's a bottleneck. So if you look across the maize genome, a typical gene in maize shows less diversity than that same gene in teosinte. So this by itself is not much of a signal. That's an enormous signal. You have lots of variation in teosinte, you've got no variation in maize. Now, a couple of interesting things. Where's that signal? It's not in the coding region. It's in the non-coding region. Furthermore, we now know where this signal is. It's 64 kb up here in a retrotransposon called hopscotch. And we can tell that that jumped in it was about 30,000 years ago. How can we tell? When you have a retrotransposon that jumps into a DNA, it makes staggered flanking cuts. So when it's inserted, it's got identical repeat sequences around it at the point of insertion. Those sequences then can diverge, and we can crudely age the transposon by looking at the amount of divergence between those initially identical flanking sequences. So the mutation that generated TB1 is clearly under selection. I've got reduced levels of variation, and the mutation was basically caused by a regulatory element that actually occurred far upstream of the gene. So this is kind of a classic example of a domestication gene that has a strong signal being under selection because there's a really reduced level of nucleotide variation. So this is one of the classic examples of an obvious gene under selection with really lots of interesting implications on it. So selection changes the structure of the coalescent. So a sweep that is a favorable allele increased to fixation not only changes the size of the coalescent, it makes the time to most recent common ancestor shorter, but it also changes its shape. So if I would, for example, take a coalescent with a long time back to common ancestry under drift and photographically reduce it and make it smaller, it would have a certain shape, even though it's smaller. And that shape basically is the nodal lengths increase as one goes back in time. Remember, in that coalescent time, half the coalescent time is going from 2 copies back to 1 copy. Those nodal lengths, as you go back in time, get larger and larger and larger. Under a sweep, those nodes are compressed as you go back in time. It looks more like a thistle, like a star coming out of that. So not only is it shorter, you get nodal lengths being compressed. And the difference, the importance of that is, think about those geometries. If you throw mutations on those geometries and then change those geometries, you get different pattern mutations at the end. If I have a coalescent looks something like this, under drift, a mutation up there gives me mutations in those 3 copies. If my coalescence sort of looks like this, then I get a different pattern of mutations. So for example, down here, that mutation is only on 2 copies. But here, I'm going to get much more singletons because of that. So changes in that geometry of that coalescent structure, because the nodes are compressed, gives you as much of a signal as the fact that it's been shrunk. And that signal comes in the site frequency spectrum. So don't worry about the details about how that might happen. You either see it or you don't, but realize that the compression of the size gives you a signal, but also the change in geometry gives you another signal as well. So it basically shifts not only the amount of variation, but shifts its nature. So the size shrinking changes the amount of variation, the geometry shrinking changes the nature of the variation. So one way to think about that. So under a sweep, nodes are compressed. This is often called the star genealogy or a star phylogeny or a palmetto. The structure under a partial sweep and long-term balancing selection is also different from drift. So here's some kind of cartoons. And again, this is a realization. If you did this many, many times, you'd see noise in here. But so here's a neutral. Here's balancing selection. The time back to common ancestry is deeper. And you see 2 different genealogies. A neutral genealogy for one allele, a neutral genealogy for the second allele. Balancing selection keeps both alleles around, so they're neutral down here, but then they go back to the deeper common ancestor. Under a sweep, I've got a shorter time, the nodes are much more compressed. Under a partial sweep, some of the alleles have swept through or undergoing selection, others haven't. So the key is, when you have selection, it not only changes the height, the size of that, but it also changes the geometry. And both changes in height and change in geometry give you differences in signal. So I talked a lot about the coalescent. Let me just point this out without going into the mathematics behind it. The book covers that. But those changes in the coalescence are what gives you signals that depart from neutrality. And you can then ask, for example, what type of signal do I get from balancing selection, from a sweep, from a partial sweep? And you can design tests to then look for those specific signals. Yeah? Great point. So these aren't variations, these are genealogies. So what it says here is these 4 alleles have a very recent common ancestor. And this allele has a deeper common ancestor back. So these alleles are being swept. They've got a recent common ancestor because they're all under selection. Therefore, maybe the mutation arose here. It very quickly increases in frequency. The genealogies have no mutations on them to show you the patterns. If I throw mutations down, so for example here, what I'll see basically is under a partial sweep, a mutation on here gives you a singleton, a mutation on here gives you 4 different copies. So I'll see basically an excess of high frequency alleles here. Because if I throw that on here, a single mutation, so if I throw a single mutation, half the time it ends up here, half the time it ends on this branch, most of the time on this branch it ends up here. That gives you a cluster of high frequency sites. Over here, a single mutation, maybe 2 copies, right? It's a bit different. The geometry gives you different configurations. And so with the partial sweep, you tend to get more high frequency derived alleles than you expect under the Waterston distribution. Quite a bit different than quantitative genetics, huh? But again, we're looking now at not at traits, but looking at alleles. So that's why I put this course together, is you can take courses or read about that'll tell you about testing for traits, testing using just molecules, or ecology of relating traits and fitness. I've never seen a course that sort of puts all these together. And because of that, you get these really weird different views of selection. So I wanted to try to do that to show you what the connection between all these things. But it requires covering a lot of different fields to do that. Yeah. Yeah. Uh-huh. Yes, let me tell you. Great questions. Listen, what does this actually mean? What this means is that these here are the alleles that the sequences that I sample. So if I look, for example, on a spot on the chromosome, this is the pattern, the age patterns for how those alleles are connected. Then I go to some other site. And some other site may be under selective sweep, so it's got much more recent time, so it's got less variation. And it's also got a different pattern of variation. So this pattern, when we talk about sweeps, the length of that pattern depends upon the trade-off between selection and recombination. If think about time. The longer the time, the more recombination. If I've got a site and it sweeps through very quickly, selection is strong, it's going to influence a large region because there's not time for recombination to break it up. If I've got a weak site that moves through slowly, there's lots of time to break it up, and it's only in a small region about that site I get a signal. So we'll talk about S over C, selection over recombination, sets the width of a sweep. And it's a trade-off between if I've got strong selection and low recombination, it's going to influence a big chunk of my chromosome. If I've got weak selection and or strong recombination, it's going to influence a tiny chunk. So these are not features of the entire chromosome, much less the entire genome, but these are features in a specific region. And the size of that region depends upon the trade-off between the strength of selection and recombination. A really good question.
So you could reduce this to like a single SNP?
Sure. So for example, this says, okay, in a SNP on this gene, when I look at the SNP, what's the pattern back in time? Here they are. Yeah. The coalescent takes a while, but it's a really powerful way to think about things. In fact, coalescent theory is kind of, given a much stronger way to look at drift and molecular populations. And there's a couple of very good books on the coalescent theory you can get out now. Just Google coalescent theory. And Whiff's book is very good. And then I can picture his face now, I can't think of it. At Harvard has a really nice book on coalescent theories. Wakely has got a nice book on coalescent theory. Sorry, I heard ciphering. So great question. So what does that do to the psi frequency spectrum? And the answer is, if I take that genealogy and throw mutations down, it shows me what I'm going to see. So think about having one mutation. Over here, if I put that one mutation down, half the time it's on this branch, I get one derived allele. Half the time it's on this branch, and it'll mostly be in here, that gives me 4 derived alleles. If I take that one mutant and throw it down here, and again, it gets thrown down randomly with respect to the length, I get 2 derived alleles, tiny time of getting 3, 1 derived allele, 2 derived alleles. So I get a very different pattern based upon, given that genealogy, how I throw mutations down. And that's a powerful way to think about coalescence. The drift structure or the genealogy gives you that pattern. Then you throw mutations on top of that. And those two together give you the site frequency spectrum. So sweeps come in many different flavors. And an important realization was the idea that we can have hard sweeps and soft sweeps. So a hard sweep is when a single favorable new mutation arises and immediately is favored by selection. So it's on a single background, it drags that background up. This is where you get a clear, obvious signal. A lot of tests are built around hard sweeps. That may not be the most common fashion we see. A soft sweep is when a mutation appears and selection says, who cares? It kind of drifts around, or it's slightly deleterious, then the environment changes, boom, then it takes off. Because it's drifted around before it's under selection, it may now be on several different haplotypes. Show a picture in a second. That results in multiple haplotypes being fixed and therefore giving you the first pattern. That's called the single origins soft sweep. The other thing is, you could have multiple mutations that arise during a sweep that are all favorable. And again, that spreads the signal out over several different haplotypes, gives you a multiple origin soft sweep. The bottom line is soft sweeps give you less of a signal than hard sweeps, and a lot of the classic tests that use hard sweep signals will not detect a soft sweep. So here's the kind of the cartoon. Here's a hard sweep. This is a chromosome. The box is the favorable mutation. It arises, boom, it's automatically favored. A good example, Biston bechellaria, or as my college roommate used to call it, Bitchin bechellaria. Biston bechellaria is the pepper moth. It's actually found throughout the US, and it was the dark geometrid moth that had originally was white, and metallic forms started appearing in England during the Industrial Revolution. We can actually date, there's a nice Nature paper that dates the creation of that, again, a mobile element insertion, right around 1840. That's when the mutations first started taking off. That's probably a hard sweep. So when that melanic mutant appeared, it was automatically favored and it sweeps through the population. And because it's on a single haplotype, that haplotype is fixed. How long that haplotype is depends upon the strength of selection relative to recombination. If selection is strong, that haplotype that's fixed is large. If selection is weak, that haplotype that's fixed has a small length. Contrast that with a soft sweep. In a soft sweep, a mutation arises and it's neutral, or if it's deleterious, it's only mildly deleterious. So it hangs around. If it's really deleterious, it gets removed and it has to wait to reappear until the population makes it favorable. So because it's around, it drifts around. If it drifts around long enough, it's now on several different backgrounds. Now, at this point, selection favors, And so now I see 3 different haplotypes. There's a collection are fixed, and I see a very different signal. Here I see almost no variation. Here I see variation. And how much variation depends. So I could actually have a fixed soft sweep that really gives me no significant reduction in my level of polymorphism. So I wouldn't see it with an obvious signal. So the realization that soft sweeps are out there, either a single mutation arises and eventually gets on different haplotypes before favored, or it could be these are independent mutations which all arise and are all favored on different haplotypes. And soft sweeps basically give you a much weaker signal in most of the features than hard sweeps do. So tests are biased for hard sweeps. The realization has made people look for tests for soft sweeps, but again, the vast majority of tests which are out there are basically hard sweep type tests. So what about the population genetics of a sweep? I've given you the key idea. The key idea, it's a race. I have a site that's now favored. It goes to fixation. The speed at which it goes to fixation depends upon the strength of selection. So speed is kind of 1 over S. If S is small, it takes a while. But remember, if S is bigger than 1 over 2n, it still changes faster than drift. Likewise, the width of that depends upon the recombination fraction. So I have a site here. So the computer is where, so let's imagine the computer here is where the favorable allele occurs. Here is a mutation. I'm at some recombination distance C. Here's another site. I'm at some further recombination C. Assume it's neutral. What is the impact of selection there on this site? It obviously depends upon the recombination rate and selection on that site. If this site goes to fixation very quickly, there's very little time for recombination to act, and this site gets dragged along with it. This site over here, further away, if that size goes quickly, I've got a much larger C. I can have some decoupling, whereas in this case, for example, if there's essentially no recombination, this thing is fixed. As recombination and selection change, then the fraction that's fixed goes from 1 to something called f of s. This is the fraction that's fixed. And as you'll see here, hs basically is the strength of selection in the heterozygote. So what fraction here depends upon the initial frequency of the favorable allele, the recombination frequency, and the strength of selection. And this is the classic result here that Maynard Smith had, and this is a result here that adjusts for finite population size. So the key parameter is this fraction of the initial association that remains after a sweep. That fraction goes down as selection becomes weaker. It goes down as recombination becomes stronger. So the width of how much a sweep impacts depends upon this, and that depends upon the relative strengths of recombination to selection. A good example is if you look at a highly self-species like rice. Recombination only effectively occurs in heterozygotes. It occurs in homozygotes. It doesn't change anything. You're just swapping identical DNA strands. What happens in a highly inbred species? Heterozygosity goes down. What it means is the effective recombination rate goes down. So a sweep in rice can, in fact, megabases of DNA, 100, 200 megabases of DNA. A sweep of the same strength in an outbred crop activates a tiny region. Why? Because effective recombination is much less in rice, and so the effective value of C is much lower. So this key parameter here basically tells you how wide the sweep is influenced, and depends upon the recombination fraction and depends upon the strength of selection. Stronger selection, larger F. Weaker recombination, larger F. The ratio of those basically give you the width of how that reaction can occur. So we can do a couple things. We can ask, what's the expected change in a neutral allele frequency? So if Q is the frequency of a neutral allele A, then its total frequency shift is given by 1 minus Q f of s. So if f of s is 1, it means it's completely dragged on, and you go from a frequency of Q to a frequency of 1. So what you see here is the impact then of the sweep comes in this term here, and again, that's a function of the ratio of recombination divided by selection. So what's the final allele frequency following the shift? Well, if S is 1, its frequency is 1. If F is less than 1, it becomes less than that. So a partial sweep will shift the allele frequency, but F of S here will be less than 1. So this general measure here basically tells you about what the strength of selection is acting on a, what the impact of a sweep is on a neutral allele some distance away. So as I'm close, that allele is almost always fixed. As I get further away, that allele is not fixed, but it's dragged to higher frequency. And eventually we'll get far enough away that the impact of that sweep will have very little impact on that neutral allele. So this thing here is really one of the key parameters in determining the strength of a sweep. So an obvious question is, okay, what's the impact of a sweep on heterozygosity? So Dick Hudson and Norman Kaplan, no, I thought I had a picture there, had basically a rough rule, and the book gives more exact expressions, that sites within 0.01 S over C show a significant reduction. That's on both sides. So the width of the reduction is going to basically be 0.02 times C over S. So for example, some simple numbers. If I look at a sweep and it covers 10 kilobases, so I see a 10 kilobase reduction in levels of polymorphism, I want to ask how strong is the sweep. If I know the recombination rate in that region, I can do that. If the recombination rate basically is 2 centimorgans per megabase, that is a 2% recombination rate per million bases, it's a little high figure. Usually it's about 1, but if that's the case, then I can plug it in here, and the strength of selection was about 5%. You can get more accurate measurements on there, but the bottom line is you can actually use that amount of reduction in polymorphism, if you know the average recombination rate, to actually estimate the strength of the sweep. Kind of cool. The other thing is these pictures here are misleading. Let's imagine you see a sweep with a big valley like that. What's your obvious assumption? Oh, that's the side of the sweep right there. So if you see a trough in variation, people usually take the middle or the low point as being a good indicator of where the sweep side is. It turns out when you do simulations, that's not the case, because random recombinations occurring early on can really make it very asymmetrical. So when you see a trough to variation, you can't just assume the middle or the low point corresponds to the sweep sites. It doesn't just work out. So just heads up on that. And again, the book gives you some more references about that. You can take this more exact expression here and have some fun with it. So you can basically say, suppose I've got the following. I've got a allele that has a 1% effect, effective population size of 1 million, and a recombination rate of 1% per million bases. That's a pretty typical value. Humans have a total map length of about 3,000 centimorgans, and we've got 3 gigabases of DNA. That's roughly that. So if I take and ask, what's the reduction? That is, if I take the heterozygosity after sweep and the initial heterozygosity, if I have a dominant allele and I'm 1 kilobase away, I've got only 1% of the initial heterozygosity. If I'm additive, I got 2%. If I'm recessive, I've got almost 20%. Why? A recessive allele takes a longer time to sweep, right? Faster you go to fixation, longer region of influence. It takes you longer to go, and recessive alleles take longer because you have to wait till you're a homozygote to have any figure. They take longer, therefore they have a weaker signal. You go out 10 kilobases, here you only have 10% of the heterozygosity in dominant, 20% of the initial in recessive, but you've got almost 70%, sorry, in an additive, almost 70% in recessive. So when you have recessive alleles, the reduction in polymorphism extends over a much smaller window. Again, time. Recessive alleles take longer to become fixed, therefore there's more time for recombination to act. What you basically see here is for a lot of the range, additive has about twice the value of dominance. Again, exact expressions are given there. So that's during the sweep. When a sweep ends, you start to recover variation. And when I was working through this, one of the things that that I was really surprised by is you would think that the recovery variation depends upon the mutation rate, right? It doesn't. It depends entirely on the affected population size. So basically, here is the recovery. So if a sweep has occurred T generations ago, what's the existing level of variation? There's an expression for it, and the key feature here is this part here, that's the reduction when the sweep is completed. This is the decay in that. You notice what doesn't appear in here is the mutation rate. So for example, on average, after about 1.4 NE generations, you recover about half the variation. And the reason for this is that equilibrium level of variation is set by the mutation rate. So if you have a tiny amount to begin with, you don't lose that much, but you don't have to recover that much to begin with. So mutation rate actually doesn't appear in rates of recovery following a sweep. So if you read about people looking at microsatellites, which have very high mutation rates, and they notice that the level of variation seems to be much higher in microsatellites, and they argue that's because microsatellites have a higher mutation rate and therefore recover from a sweep quicker, that's incorrect. The reason you get the higher levels there in microsatellites is because mutations are occurring while the sweep is in progress and generate them. So the key feature here, though, is the sweep, in terms of loss of polymorphism, has a fleeting signal. And that signal is on the order of the affected population size. So just because the sweep has occurred, if you don't catch it soon enough, its signal is gone. And that's, you might obviously expect that, that's a feature of all these different tests. And the key feature is, over what time interval do these tests work? So not only do we have tests that work for hard sweeps and not soft sweeps or vice versa, we also have tests in different intervals. There's some tests which are very good at catching a hard sweep while it's happening, but no good when it's almost done or done. We have tests that really can't catch a hard sweep as it's happening, but are really good detecting it once it has occurred. So we have different types of tests for different types of sweeps and different types of time signals over where we can detect different types of these things, which is why no one test fits all. That's why the literature is so convoluted, is there's all these different approaches to try to pick out these different signals. And again, we're going to cover all those over the next couple of days. So questions so far. We talked about sweeps and how it perturbs the level of polymorphism. Uh-huh. Right. No, it's actually a proxy for drift, basically recovering it and getting genealogies back together again. So so remember, you have a certain level of variation. That's a function of mutation, pumping in variation, and drift removing it. You've removed variation based upon the sweep. Now what's happening is you're removing that original signal because you're shifting up the coalescence in that. And that, so the point about it is this: if I have low mutation to start with, I have a slight reduction in my heterozygosity, so it takes a certain amount of time to go back up. But I have low mutation, so it takes a while. If I got a high thing, I've got a big jump, but it takes a certain amount of time to go back up, and that depends upon the mutation rate. Mutation rate basically cancels. So it really only depends upon the affected population size and the time since the event has happened. Because remember, you're getting to some equilibrium level, and you're assuming the mutation rate hasn't changed. So that equilibrium level takes a while to get to, and it's at same level because of the strength of selection. How much you have to recover depends upon how long you have back to common ancestry. It's an odd result. When I first saw it, I was surprised people hadn't pointed it out before. If you look, people actually mentioned it, but it's kind of buried in some of the literature. Yeah? I'll give an example of that, actually. Yeah, so multiple origins. So an example would be if I've got a population over geographic structure. And a good example would be the ability to utilize lactose as an adult. There have been independent mutants for that over a geographical space. So it's not the case that it's a single event, but rather it's multiple independent origins have generated that. I'll give an insecticide example where you basically have to have multiple mutations to see something like that. And the point is, if your we'll talk a lot about that a bit later on, if your population size is big enough, the chance of having multiple mutations is much more than you might think. And those mutations occur while your one allele is being driven to fixation. These will jump in and give you a diverser pool of haplotypes, pardon me, as you go ahead and do that. Uh-huh. I'll have to think about that. Yeah, I think trying to think of the softening of sweep. I think you're right. Yeah, these results here are, once the sweep is done, you're assuming you have a constant effective population size. A big problem with a number of these tests is something called, it's a great name, allelic surfing. And allelic surfing is, think about humans. Humans had a wave of population growth coming out of Africa. And as we'll talk about, the idea about correcting tests for population structure is that if you have common population structure or common demography, it'll influence every site in the genome equally. That's true if you have a population that's static in space. If you have a population that's expanding in space, a new mutation on the edge of that expanding wave can give a coalescent structure that looks very, very, very much like a sweep. And that signal would be against an otherwise background because that allelic surfing doesn't affect all sites in the genome equally. So I think allelic surfing is a huge issue for correcting for population structure because most populations people are interested in have expanded, so therefore the environment's changed, therefore there's lots of selection, but they also have got this expanding population structure. And so things like allelic surfing, where mutations arising on the edge of this expanding wave have a coalescent that looks quite a bit like a sweep. It's actually quite hard to tell those apart. So a big caveat, if I were a really mean, vindictive reviewer, every paper that comes across me for sweeps I could say, there's no evidence this just simply isn't allelic surfing. And I could be pretty reasonable about doing that as well. So the bottom line is, tests of selection people are really excited about them. There's lots of fun, but there are issues with using any of them. Just be aware of those issues. Uh-huh. Not with any power that I'm aware of, which is kind of sobering. But then that's the optimist pessimist, right? The optimist, you can go, well, they're kind of rare, so my site must be under selection because it's my site. I care about that. Whereas the reviewer could go, I don't care that it's your site. You haven't shown that it's not surfing. And we'll give a couple of examples a bit later on, classic human examples, which for all the world look like selection, which are probably surfing. So on that sober note, just be aware of that. So I want to talk about then, we talked about, this is the Waterson site frequency spectrum. That's the expected value of seeing a site with exactly I copies under equilibrium neutrality. How is that shifted by a hard sweep? And here's how it's shifted. Waterson distribution is given in black. The blue is the distribution. So number one, you see the Waterson distribution, a lot of rare alleles, very few common derived alleles. This is unfolded. What you see here is it sweeps it down to low, and you can basically look at the structure from basically here. So a fraction of sites get shifted to low frequency, a fraction gets shifted to high frequency. So you get this structure here. You get an excess of low frequency derived, and this is a big signal, an excess of high frequency derived. This is an unusual signal you don't expect under most population structures. You do expect it under surfing potentially. So if you have the unfolded spectrum, an excess of low frequency and high frequency derived. If you have the folded spectrum, these get folded into here, and you see an excess of minor allele frequencies. But this is a big signal. That's an unusual signal that most tomographies won't give. So an excess of high frequency alleles is a good signal for a sweep. The problem is it's very fleeting. It goes away very quickly. So excess of sites with high frequency derived alleles, or more generally, an excess of site with rare alleles if you fold the frequency spectrum. Now that assumes you start with a Watterson. That's an equilibrium model. If you're not at the equilibrium model, you don't start with a Watterson and you perturb some other distribution. And there are tests that actually take the so-called empirical site frequency spectrum, that is, take sites you think are neutral away from your target site, look at what the frequency spectrum is at those sites, then figure out how they would be perturbed with selection, and then ask if your data fit that non-equilibrium site frequency spectrum that's now been perturbed. LD is impacted in very non-intuitive ways by a sweep. So initially, a sweep generates lots of LD, and so a strong signal for an ongoing selection is what are called long haplotypes. However, as the allele gets fixed, you've got this weird feature of little LD at the selective site, because it's removed most of the variation, but lots of LD on either side. Here's kind of the cartoon. So here is our sweep site. Basically, these are distance from the sweep site. Here's the beginning of the sweep, the end of the sweep. What you see is as the sweep goes on, you get a big pulse of LD right around the selective site. And then as the selective site becomes more common, you then get this trough in the middle with LD on either side. Very non-intuitive. And the net result for that is that you get different signals depending upon when in the sweep you are and depending upon what type of sweep you have. This is for a hard sweep. A soft sweep generates a different pattern of LD, and we'll talk about that a little bit later. But the bottom line is, besides site frequency spectrum, you can get signals for a sweep looking at the LD pattern. Yeah? Sure. So LD basically linkage to equilibrium, and it simply means if I take two alleles on different loci, they're correlated. So for example, if one allele has frequency 0.5, the other allele, so the coca allele has frequency 0.5, and the pointer allele, my phone allele, has frequency 0.3. If you have the coca allele, you have a different frequency than 0.3 of having this allele. They're either positive D, that is, they occur more frequently together than you expect, or negatively, they occur less frequently together than you expect. So it's a correlation of alleles across different loci, and that generates patterns we can use. Yeah? Part of the reason is because LD is a function of allele frequency. If you've got no segregating alleles. And the other reason is early on that coalescent, a recombination or two can give you very different asymmetric features. The book gives you references into some of the papers about this. It's kind of esoteric, and it's one of those things that I think I understand, but I'm not. I think I'm 98% sure I get it, but there's always that 2% out there. So I'd always recommend read the original papers. Not saying they're right either, but LD does weird things about that.
So would you expect this pattern in allele as well, as it's a function of allele frequencies, or
Boy, so allele frequency would distort the coalescent. That's the problem. You have to figure out how that distorting of the coalescent is going to translate into here. I don't have a good feel for that. That's a good research topic for you. And that's not meant, by the way, to discourage questions, assigning you as a research topic. So don't let that defer people from asking questions. So what are the summary of a hard sweep signal? An excess of sites with rare alleles in either the folded or unfolded frequency spectrum, an excess of sites with high frequency derived alleles in the unfolded spectrum, a depression of genetic variation often asymmetrically about the type. So that's what an ongoing or recent sweep. What about the LD patterns? When allele is at moderate frequencies, that is, the favored allele is still being swept, it's a partial sweep, it either could stop or it's ongoing, you see an excess of LD surrounding the region, long haplotypes, we'll talk about. Following fixation, you see this odd feature of an excess of LD on either side, but a depression of LD around the site. And there's a test for that we'll talk about as well. Finally, signatures of a sweep, a hard sweep, are very fleeting. So remaining on the order of about a half NE for the excess of rare alleles, 0.4 NE for the excess of high frequency alleles. Number 3 here is about 1.4 NE, and down here is about 0.1 NE. So the bottom line is these are all in the order of between a tenth and NE. So, and this is a hard sweep. So again, you've got this issue of time scales and types of sweeps, and it's why if you pick a particular test, you're going to miss most selection that's ongoing. So what we're talking about here basically are tests for a single ongoing or recently completed event. On Wednesday, we'll talk about things like MK and HKA tests, which basically look for multiple adaptive substitutions over a long period of time. These tests miss that. Likewise, in those tests, there could have been a single adaptive substitution, which is phenomenal. We picked up here, but we wouldn't pick up on a test like that. So most selection events are missed by these tests. And that, I think, is a good place for a lunch break. So we're talking about sweeps. And some of the big concepts we've talked about are when you have a sweep, it changes the coalescence structure. It not only changes its length, but it changes its topology, its geometry. And those are both signals that different tests pick up. We also talked about how linkage disequilibrium is generated during the first part of a sweep, and then it has an unusual structure at the end of sweeps. Those are also things that we'll talk about. Then we talked about soft sweeps, either single or multiple origins, where the initial mutation under a hard sweep, once it appears, is immediately under selection. Under a soft sweep, it either drifts around a while or you have multiple new mutations that arise when the sweep is occurring. That's kind of where we're left at this point. We'll talk about multiple origin soft sweep with the recurrent mutations in a second, but as always, let me see if there are questions about anything except colonoscopies, but you know. That video just kind of, okay. So under a soft sweep, a single origin, a mutation arises and kind of hangs around a while before it's under selection. The other option you can have is that several new favorable mutations can arise during the event. So you have a mutation. We usually think they're unique, but they don't have to be. Likewise, you could have mutations in several different spots arise. So it may be in the same gene, but instead of being the same mutation, maybe it's another mutation in that gene. Good example are knockouts, right? If you have a gene where the new phenotype now is that gene gets turned off, there are multiple ways to knock out a gene. You can have a multiple origin soft sweep by having multiple knockout mutations arise and as a group get fixed together. So if you've got multiple mutations that arises, then selection fixes a set containing multiple haplotypes, again leaving a weak signal. And so how can that occur? How likely? The key is theta sub b, which is 4NE mu, and the b there means the mutation rate is now the beneficial mutation rate. So if this quantity is tiny, for example 1%, multiple origin soft sweeps are very rare. If it's a little bit less than 1, they're intermediate. If they're bigger than 1, they're almost certain. So an example of this, and it gets back to one of the other questions here, was this data coming out of Stanford about Drosophila melanogaster, the ACE gene, which is in the acetylcholinesterase, is the target for many commonly used insecticides. So an obvious thing to look at is, okay, you look at when the insecticides were introduced, and you ask when these mutations arose. And single nucleotide changes at 4 conserved sites convey partial resistance. If you have 2 of these changes, or 3 or 4, you have increasing levels of resistance. And so the major insecticide use that this targeted started around the 1950s. The authors found that mutations in both North America and Australia had risen de novo following the migration of melanogaster out of Africa. Remember, melanogaster is commensal. It followed humans out of Africa. Given that only about 1,000 generations have, elapsed since this gene became a target, and estimates based upon nucleotide diversity give you a theta about 0.01, and that means a theta about 1/3 of that. Why? Because only one of the 4 mutations can be consistent. Then you wouldn't expect to get these mutations that show, these sequences that show 3 or 4 of these mutations. The issue is this data here based upon nucleotide diversity is based upon the long-term effective population size. However, if the effective population size in the last 1,000 generations or so was closer to the census size, then you get theta be equal to 1, and these multiple origin mutations were quite likely. So one of the concerns the authors had was, how can you reconcile levels of nucleotide diversity which suggest a tiny value of theta, and again you can adjust that for what it should be given the beneficial, with the fact that you do find double and triple mutations in natural populations. And those require multiple events, and those seem to be quite unlikely. And the answer is, this nucleotide diversity estimate of theta there is based upon a long-term historical effective population size. If your effective population size was significantly larger in the period where you're looking for these multiple mutations, you could indeed see those multiple mutations. So effective population size based upon nucleotide diversity is a long-term historical average, and your window for effective population size might be quite different. So the bottom line here is an example of a mutation, of an adaptation that took multiple rare mutations simultaneously to occur. That seems unlikely on the face of it, but that's because you're using historical long-term averages and not the averages you had over this time window here. So what are the signatures of a soft sweep? Either multiple origins or a single origin soft sweep. Well, there's very little reduction in H under many soft sweeps, so H is not a good signal. H, the heterozygosity, the nucleotide diversity. The key is LD is a good signal. Under a soft sweep, LS, it should be LD, fix that right now before I forget. Under a soft sweep, the linkage disequilibrium signal extends through the site. So we'll talk about, so remember, a hard sweep, the LD signal is on either side but not through a site. In soft sweep, it is through a site, and there are different tests we'll talk about that distinguish hard versus soft sweep using the LD signal. There's no one linkage disequilibrium test that does both. You have to use the 2 different ones, because one detects hard but not soft, one detects soft but not hard. Then the biggest concern is what's called the polygenic sweep. So remind you, a hard sweep, a new mutation arises and sweeps through the population. A partial sweep, a mutation is increasing in frequency. It hasn't become fixed yet. It's got 3 possible fates. Number 1, it's on its way to fixation. Ultimate fixation. Number 2, it's on its way to some equilibrium value. So I've got, let's say, balancing selection, and my equilibrium frequency is 1/2. This thing moves from 2% to 40%. It's not going to go to 1, but it's increasing in that. The third possibility is the environment caused it a little to be favored, its frequency changed, then the environment changes. So it's not reaching a selective equilibrium value. That's a partial sweep. That'll generate a certain type of signal. Then we talked about soft sweeps. The third type of sweep is a polygenic sweep. And here I've got 2 possibilities. Number 1, fixation over a large number of sites. So the alleles go to fixation, but each site has a small effect. Number 2, small allele frequency shifts over a number of sites. So either a small collection of partial sweeps or a small collection of hard sweeps. But on any particular trait, the idea is the trait's determined by a large number of loci, each of which has a relatively small effect on the phenotype. That means that S on any given locus is tiny. We can actually work out, for example, if S is 0.1%, then if you take a typical recombination rate of 1% per million bases, a sweep encompasses roughly 2,000 bases. So you get a depressed region around 2,000 bases. That's not a lot. Human diversity is about 4 or 5 polymorphisms in there. So that's a small window unless you have a very highly polymorphic species. And that's the best case of a hard sweep where these things all go to fixation. More generally, a sweep could involve relatively small allele frequency changes over a large number of loci. Small partial sweeps over a large number of sites wouldn't be much of a classical signal there. Now, one of the things we'll talk about later on is we might find correlations in allele frequencies when different populations are sampled over similar environments. This is one of the original tests that was used, was they basically took different populations. They basically took alleles and then sampled over human populations and asked if there was a correlation, for example, between UV levels and allele frequencies. So the environment over independent sites was generating correlations. We'll talk about some of those tests a bit later on. So hard sweeps leave the best signal. That signal is fleeting. Soft sweeps leave special signals, but only using specific tests. Those signals are weak. Partial sweeps, again, think of those as a weak sweep. It doesn't leave much of a signal. Polygenic sweep leaves the worst signal. So even if there's lots of selection occurring, there's good reasons to suspect that the method you're using is not going to detect them. So again, a key point of this course I want to reiterate over and over again is that tests for selection are very seductive, but there's no omnibus test that works for all situations. Most type of selection you're going to miss. You can either catch currently ongoing selection or stuff that has just occurred, or you can catch scenarios where there's a long-term history of repeated events. A lot of things fall in between that. So most selective events we're probably not going to detect. So just because selection is occurring doesn't mean you're guaranteed to find it, at least using the marker approaches. Okay? Yeah.
One thing that I've often heard thrown around is that all the methods that find selection on character traits,
Right.
phenotypes, assume polygenic sweeps. Does that track?
No, it doesn't. So you can, so the methods are basically look for character signals. And again, so there's a couple of methods. So a character signal simply means that that mean has shifted in a pattern that's not consistent with drift. You can have it shift in a pattern not consistent with drift if you've got one or two loci of large effects or if you have an infinitesimal model underlying them. So those usually don't necessarily track. Other questions or comments? Again, don't be bashful. Have we lost a couple of people? See a couple empty seats? That's okay. They're lost. They missed the how to enjoy colonoscopy. You can never unsee that. So we can take, so far we've talked simply about what signals do we expect to see. We can then ask a really cool question, which is, if sweeps have been occurring, how does this affect the entire genome? This is a separate issue. I'm not trying to find a specific site under selection. I'm asking, if I look at the genome, what does the genome tell me about selection in general? How common is selection? And again, we have two types of selection. Selection that everybody acknowledges is always occurring. Mutations arise, most are bad, they get removed. We call that purifying selection. We kind of don't care about that, because if you find evidence of that, it's like, that's not a publishable result, because you expect that. The type of selection people want is adaptive evolution. A mutation arises that either immediately or eventually becomes favorable. That favorable mutation then increases in frequency in the population. What are the consequences of selection for structuring the genome? Well, it's basically two models. Recurrent sweeps means across the genome you have episodes of favorable mutations arising and being fixed. So it's a history of adaptation. Or you've got background selection, idea due to Brian and Deborah Charlesworth. In background selection, you have deleterious mutations arise which are removed, and both recurrent sweeps and deleterious mutations result in a reduction in the effective population size at linked sites. They basically make the coalescent trees shorter And you can think about that time in a coalescence tree as one measure of effective population size. So we expect background selection to always be occurring because deleterious mutations arise and they're removed. The issue is, what's the relative importance of these two? And the bottom line is, it's extremely hard to distinguish background selection from recurrent selection. One common feature that you see in almost all genomes, almost all, is if you scan across a genome and have some measure of average recombination rate and some measure of polymorphism, what you see is there's an inverse correlation. Regions with low recombination, sorry, positive correlation. Regions with low recombination rate have low levels of polymorphism. What are classic examples of regions with low recombination rate? Telomeres of chromosomes have reduced recombination. Centromeres around chromosomes have reduced recombination. So remember when I showed you that slide for a GWAS with all the little pixel points? Some of you might have noticed that there are white streaks where there are no pixels in that slide. Those white streaks correspond to centromeres, because around centromeres, there's almost no SNP variation. So it's a pretty general feature that for whatever reason, when you have regions of low recombination, you tend to have low levels of polymorphism. Now, one explanation is maybe mutation and recombination are inversely related. So if you don't get a lot of recombination, you have lower mutation rates. That was quickly dispelled because you can look at divergence and see that sites and telomeres or other low recombination regions diverge about as average as sites elsewhere. If they had reduced recombination rate, you'd also expect them to have less divergence. You don't see that. So there's some feature that's giving you these reduction in polymorphisms in regions of low recombination. And the two obvious explanations are both based on selection. Number one, low recombination, you have a selective sweep because it's low recombination. You've got a longer region that it covers, so you basically have a larger region influenced, so even rare mutations can sweep that region out. Number two is background selection. With background selection, again, a region of low recombination, you've got lots of sites that can be deleterious, and either one of those will reduce the effective population size and basically give you lower levels of variation. So one clear signal we have that selection is important in structuring genomes is this pretty common pattern that polymorphism is reduced in regions of low recombination. So you can work out, and Wolkheim-Stefan was one that worked out a bunch of this stuff. You can look at what the level of diversity would be without a sweep and what it would be with recurrent sweeps. So some simple formulas you can get. So here's one formula. So rho here is basically the average recombination rate. Where do I say that? And you can basically look at the observed level of polymorphism measured by nucleotide diversity and the level without sweeps and get this expression that depends upon the rates of recombination and then depends upon, per nucleotide recombination rate rho. This thing, gamma, is basically the scale strength of selection. And this thing here, lambda, is the per nucleotide rate of adaptive substitution. This seems to be kind of a home run thing to get at, but as we'll see, you can actually estimate this for using a couple of different methods we'll talk about probably on Wednesday. So what this basically shows is it basically shows what the levels of reduction should be, and you should scale with 1 over the recombination rate. The problem is, if you work out background selection, you get a result that's essentially identical to this in certain ranges of parameters. So simply looking at the reduction in polymorphism can't distinguish between recurrent sweeps and background selection. And that's a pretty big deal, because if the reduction is from recurrent sweeps, it means adaptive evolution is occurring all the time. If it's mainly background sweeps, it means most of the selection is cleaning up the mess caused by mutations. Fundamentally different worldviews about what's going on, and both of them leave the same signature in the genome. If this is recurrent sweeps, one of the debates is, if I have a chromosome and that chromosome shows reduced levels of variation, is that due to a few large events? So I have a rare event that has a really high selection coefficient, therefore it's got a really big range. Or is it due to many events which are more common, but the individual sweeps are smaller? So when I see a region of depressed variation, if it's due to recurrent sweeps, is it largely due to a few big events or a lot of tiny events? And the problem is, look at this expression here. These are fully confounded. Lambda is the rate at which these things occur, and gamma is the strength of those. So you can have that value be the same value by having a few big events, gamma big, lambda rare, or gamma small, lambda big, having a lot of common things. So this result by itself doesn't distinguish that. We'll talk a little bit later on Wednesday, and also in chapters 8 and 10, about how to get separate estimates of gamma and lambda. And that's one way to get a handle on that. We'll talk about that. And here's a summary table where people have actually tried to estimate these. So here is estimates on melanogaster of this combined frequency of sweeps times the effect of sweeps. Here are where you could break out estimates of the frequency from the strength. So these sweeps here are large effect sweeps, which are rare. These things here are small effect sweeps, which are a bit more common. And notice something interesting. In Drosophila melanogaster, and most of this I think is on the X chromosome, if you look at these things where people have gotten separate estimates of these results are relatively constant, but you see dramatic differences between average strength of selection and frequency of events. So the Drosophila data seem to suggest either big and rare or small and common, and the data are very ambiguous because some suggest one, some suggest the other. As we talk about in chapter 8, this may be a consequence of the size of window you're looking at. If you're looking at large windows, you tend to estimate large selection coefficients. You look at small windows, you tend to estimate small. And some of these data we're looking at, even though it's similar data, looking at different sized windows of polymorphisms. Okay. So what you can do then is ask what are some of the consequences of these. So here, X-linked genes in Drosophila. So take these two estimates here, Aldefano and Jensen. It gives you a per nucleotide rate per generation of adaptation, either about 8 times 10 to the minus 10 or 4 times 10 to the minus 11. If I have a region of 100 kilobases, so multiply this by 10 to the 5 for Aldefanos, it suggests one sweep in a region this size every 13,000 generations. If you use a slightly larger, slightly smaller value, it suggests one sweep in a region of 100 kb about every 200,000 generations. So what I want to stress is if you have estimates of either the strength of selection, gamma, or lambda, the frequency of sweeps, you could ask a bunch of things. We now have the ability to get some crude estimates of these, and that'll tell us a lot about the average type of selection acting on a genome. So for example, 100 kb region, random 100 kb region, having a sweep every 13,000 generations, that's going to leave a pretty big signal versus under a lower rate, having a sweep about every 200,000. And the difference here is this is a lot of small sweeps. These are rare big sweeps. These are two estimates I'm looking at. And here I get small frequent sweeps. Here I get large rare sweeps. So the key is, do you get many weak versus a few strong sweeps? And again, this is something that's still ongoing. There's debate about this. There are estimates on here. And usually when you estimate, estimates give you a fairly common value for this joint product, but differ quite widely in the strength of selection versus the rate. These estimates tend to be relatively constant in terms of the total effect, but these estimates, strength of selection of an individual event and frequency, turn to have a bit more variance in those. So here's kind of the picture. Here is the impact of a small sweep. This was fairly recently. A large sweep, and then a fairly ancient large sweep. The ancient large sweep means you basically are recovering variation on here. So what are the consequences then of frequent sweeps or background selection? Well, we can look at substitution rates. So a decrease in effective population size, what does that mean? It means advantageous mutations some fraction become more neutral, it means that some fraction of deleterious mutations also become more neutral. Remember, for NES bigger than 1 in absolute value is when selection dominates. So if I have a range of selection coefficients, that range where selection dominates may be everything outside this region, so pretty big. As my effective population size gets smaller, I have a larger region of that distribution of selection coefficients where things are effectively neutral. So what are the consequences of that? Well, number one, a decrease in the effective population size leads to a decrease in the amount of polymorphism, remember 4 NE mu. It leads to an increase in the substitution rate if you believe that most mutations are deleterious, because remember, if I decrease the effective population size, it means that a higher fraction of deleterious mutations now behave as if they're neutral. It also means a higher fraction of advantageous mutations behave as if they're neutral. If the second category is pretty rare, shrinking the amount that are favored by selection doesn't have much of an impact. So if most of the mutations are deleterious, as I decrease effective population size, I should increase this. Since both recurrent selection and background selection have bigger impacts in regions of lower recombination, what patterns are actually seen in the genome? So let's look at Drosophila. Melanogaster versus Yakuba. What we see is an increase in the synonymous substitution rates on chromosomes, 4th chromosome, dot chromosome, with no recombination. If you compare these chromosomes with no recombination with the autosomes, you see they have higher replacement rates. So both these are consistent. The smaller chromosomes with no recombination would have reduced effective population size under both sweeps and under background selection. A consequence of that is higher replacement rates, because some of those mutations which were deleterious under a larger effective population size now become neutral, therefore have a higher chance of being fixed. The synonymous substitution rate basically means that you have weaker selection on these. Because of codon bias, some synonymous substitutions actually are slightly deleterious. If you look in human chimpanzees, you have no obvious effect. But one reason is effective population sizes on autosomes here are probably quite large. Effective population sizes on autosomes here are a little bit smaller. So the percent reduction in effective population size is much more dramatic in Drosophila autosomes versus no recombination versus in humans where there's lower effective population size to begin with and not as much variation. Well, another thing would be, what about correlation between the divergence rate and polymorphism? So sites with higher divergence for replacement sites may have experienced more sweeps. If they've experienced more sweeps, you expect them to have lower recombination rate and therefore lower levels of polymorphism. So a negative correlation between sites with higher rates having lower levels of polymorphism is seen in several species of Drosophila, seen in aspen, and also seen in humans. So one of the patterns you expect from from recurrent sweeps, this negative correlation you see. However, under background selection, sites with low divergence should also have lower levels of polymorphism. Why? Low divergence means stronger constraints. Stronger constraints means that more sites, more mutations tend to be deleterious. That means a higher amount of background selection. So you have a pattern here that's consistent with recurrent sweeps, but you have a pattern here that's you also see in humans of this low divergence and low polymorphism rates, which you can basically, it could arise from mutation, but you can actually adjust for that and you see this pattern. So again, the problem is we can't get a clean signal between multiple rounds of adaptive substitution giving these patterns and simple background selection giving these patterns. One other thing to look at is codon usage bias. I mentioned this briefly. Codon usage bias is what happens when I have a amino acid that's coded for by a number of synonymous codons. They all code for the same amino acid. I would expect the frequency of the synonymous codons to match the nucleotide frequency in the genome. to a first approximation. You often don't see that. You often see one of the 4 or one of the 6 synonymous substitutions, synonymous codons, being almost exclusively used. And the idea is that there's an isoacceptor tRNA for each of the different codons, and what you're seeing is a matching of the codon matches the one with the most common tRNA in the pool. And that makes translation more efficient and faster, more accurate, and that imparts a small amount of selection. That small amount of selection in a very large population size is big enough to overcome drift. In smaller population sizes, it's not big enough to overcome drift. So what can we say about that? So optimal or preferred codons should be weakly selected for, and unpreferred codons should be weakly selected against. Since S is expected to be small, a modest change in NE may leave a big signal, transforming a situation where 4 NES is bigger than 1 to where 4 NES is much less than 1. So for example, if on average I'm sitting, a typical codon is sitting right around 4 NES equals 1, if I have a reduction in the effective population size to 10%, all of a sudden that site moves from being effectively selective to effectively neutral, and I should see a difference in the codon patterns. So what can we see? So using an outgroup, that was a comparison between transitions between preferred to unpreferred, so mutations that go from preferred to unpreferred codons, with mutations that go from unpreferred to preferred. So if there's selection, we expect, if I account for mutation rates, we expect for these to be far less than those once I adjust for mutation rates. And what you saw was an excess of going from an unpreferred to preferred replacement in pseudo-obscura, that should be an A, but you saw an excess of preferred to unpreferred replacement in melanogaster. Difference is melanogaster is known to have a much smaller effective population size than pseudo-obscura. The other pattern that you might expect, pseudo-obscura, the other pattern you might expect is that preferred to unpreferred should be deleterious, and therefore in the population they'd be segregating at lower frequencies. So if you have selection that effectively acts on codon bias, you should see preferred codons occurring more frequently in terms of substitutions, and you should see mutations which would be deleterious, preferred going to unpreferred, these should be segregating at lower frequencies. And we see signals of those in some Drosophila, but not others. Why we might not see it in here? Because if 4 NES is still less than 1, that's not under selection. It's effectively neutral. So another way to look at that, so this is one of Brian Charlesworth's students. He looked at Drosophila americana and Drosophila virilis, and they observed 84 synonymous substitutions between the two species, and they classified sites as either preferred going to unpreferred. Again, preferred and unpreferred based upon codon usage frequency, and likewise looked at unpreferred going to preferred. What they saw were, here are the substitutions, here are the levels of polymorphism. These ratios, if you look at it here, the polymorphism divergent ratio is much higher for preferred going to unpreferred. So these are basically things which are, we think are weakly selected against. You'd expect fewer of those to be substituted. Therefore, you have more polymorphism relative to substitutions. These are preferred, selected for. Therefore, you should have more substitutions relative to polymorphism. And you can basically show that Fisher's exact test shows a highly significant difference. So it does matter the direction here. And the suggestion is that if you go from unpreferred to preferred, you have a higher substitution rate. If you go from preferred to unpreferred, you have a lower substitution rate. Again, that's consistent with the fact these sites may be under fairly weak selection. You can actually estimate the strength of selection. It depends upon strength of selection and the ratio of recombinations between those. When you use this expression in Drosophila, you get 4 NES is about right equal to 1. That's a magic number, because if 4 NES on average were, let's say, 100, it means a relatively small shift in the affected population size still wouldn't change that. It would still make it selected for. If 4 NES is, let's say, 10%, a reduction in the affected population size isn't going to really have much of an effect at all. You've got to be sitting right around this magic number of about 1. So on 1 population size they're selected. If I change the affected population size in the region of a chromosome, they become unselected. And Drosophila seem to be sitting, at least some species, in this magic window. And the reason that's interesting is we can then look more carefully about what codon usage bias looks like. And we see some interesting patterns. Its codon usage is less extreme in regions of low recombination. Less extreme meaning is closer to the neutral pattern. It means the impact of selection seems to be less. That's consistent with regions of low recombination having background selection and or recurrent selection. You also see less codon bias in genes that are rapidly diverging at the replacement level. Well, the rapidly diverging could be because they're undergoing sweeps. Sweeps then lower the effective population size. A cool feature is it's less extreme in long genes and the middle of long exons. That's a much more interesting pattern. How can you have codon usage bias? Now, there may be physiological reasons, but are there population genetic reasons? And so one explanation for here is if I'm in the middle of a long exon or at in a long gene, I've got a lot more sites which can mutate to deleterious sites that are nearby or advantageous sites. So in those regions, I have more of an opportunity for background selection or for recurrent selection, both of which would lower my effective population size, both of which then would basically decrease my codon bias. So there's some subtleties that get into looking at analysis of genomic data like this, and the bottom line is what's behind these short-range effects, long versus short genes, or in the middle of long exons. And an explanation is the Hill-Robertson effect. There's Bill Hill, here's Alan Robertson, both at Edinburgh. Bill was actually Alan's student. Alan Robertson is kind of like the Tesla of quantitative genetics. If you look at a random person who knows a little bit about quantitative genetics, they would say Faulkner. It's kind of like a random person on the street would say Edison was the better engineer. But people in the know it was Tesla. Robertson is the reason that Faulkner's book is so clear, because Robertson basically was at Edinburgh, went through it and cleaned it all up. Robertson is one of the sort of key figures in quantitative genetics in the last century, and Bill is probably the leading quantitative geneticist this century. So the Hill-Robertson effect was a paper they had in 1966, rather esoteric, had to do with animal breeding, and they basically showed if I have selection occurring at a linked site, that it makes selection at a nearby site less efficient. And they phrase this in terms of reduction in terms of effective population size, but there are other features as well that can be involved, such as interference. So the observation of selection at a nearby site, making selection at sites around it more neutral, that observation in general is called the Hill-Robertson effect. And this basically is what colors a lot of how we think about how genomes are structured. So small-scale Hill-Robertson effects, like within a gene, say if multiple alleles are segregating, these can interfere with the effectiveness of selection, therefore weakening. Therefore, you get a more neutral pattern. In long exons, you could have more regions which have segregating sites under selection, therefore they interfere, therefore you have a reduction in the efficiency of selection, therefore you have a more neutral pattern. So we've sort of come full circle on our review of sweeps, telling you about the signals of sweeps, and then telling you about the patterns we see in genome variation. They're due to selection at linked sites. What we don't know is if that selection is mainly for adaptation, or if that selection is mainly for background selection, removing deleterious. It's an unresolved issue, but it's actually quite important, because it basically tells us, is a lot of the selection adaptation or is it maintenance? Are you getting better or just fighting not to change? And that's kind of all I'm going to say about sweeps, to give you kind of the population genetics and to kind of close out with some of the overview. I was a bit choppy going through here. If you read chapter 8, it goes into much more detail and gives you a lot of the references so you can follow it, in more detail by yourself. So questions? Yeah, that's a good question. So clonal interference is a very comparable thing. And let me remind, so people don't know what clonal interference is. Let's imagine you have a bacteria or a mitochondria or chloroplast. You've got a circular genome with no recombination. Now let's imagine a favorable mutation arises over here. Well, you would think, okay, that chloroplast, that mitochondria, that bacterial genome is going to sweep to fixation. Not so fast. Another mutation arises over here. It's not as good as this one, but in some lineages of this, a second mutation arises. So now it has two beneficial mutations. Now it outcompetes over here. Because you've got completely linked genomes, there's no sex, you can't recombine and shift those genes together. So in asexual species, you get this competition that occurs. And often which genome wins depends upon who can accumulate the most beneficial mutations the fastest. Just getting a beneficial mutation isn't much. Those clones will interfere with each other. In a sexual population, you get recombination, putting both beneficial mutations on one genome. In asexual species, they compete with each other. Reduces their effectiveness. Other questions or comments? Yeah.
Just to clarify, you're saying that's similar to what would be happening if there were multiple organisms close proximity to one another?
Yeah, there's some commonalities but some different features. So the bottom line is if you've got multiple alleles, let's imagine the net result is, think about selection as being the difference between those fitnesses. That basically makes those alleles effectively less under selection because they're competing with each other as compared to one by itself. And that means it's more difficult. Small fitness differences can be basically swamped out by drift. And they're not nearly as efficient. The other way to think about it is something called variance in fitness. The added variance in fitness is greatly reduced, making selection much less efficient than if it was expanded. So Hill-Robertson is a couple of things, and quite frankly, it's not well understood in all its detail. Number one, it's often presented as a reduction in the effective population size, but there's additional features on top of that, having to do with non-linearities in terms of multiple locus fixation. We don't know how to solve those equations yet, so we basically frame it in terms of competition. The bottom line is Hill-Robertson simply says, if I have selection at a nearby linked site, it's going to make selection at a site, at a given site, less efficient. Less efficient means it's more neutral. And that leads back to this cryptic statement I made early on, that one of the reasons we see so much neutral evolution might be because we have so much selection going on. If there's lots of selection going on, it means a higher fraction of the mutations that appear are going to be near a linked site. Therefore, the effective selection coefficient will be less, and therefore they're likely to be more neutral. Now, if it's more neutral enough to shift it is unclear, but the bottom line is these Hill-Robertson effects can be quite important over small scales in the genome. So when people are analyzing genome sequences from a population genetic standpoint, they're almost always have Hill-Robertson in the back of their mind, which is quite ironic for a paper in the 60s on animal breeding to have such an impact on how genomes are analyzed today. Other questions or comments about sweeps? So what I've done today is I've introduced some kind of basic machinery for describing neutral variation, infinite alleles, infinite sites models, talked about number of segregating sites, nucleotide diversity, singletons. I've talked about the coalescence structure and how it has a certain geometry under drift. how that affects the site and allele frequency spectrums. Then I talked about sweeps, a bunch of stuff in sweeps, a lot of terminology, hard sweep, soft sweep, partial sweep, polygenic sweep. We talked about what a hard sweep does in terms of LD, in terms of the site frequency spectrum, shifting it, giving you a lot more rare derived alleles and an excessive number of high frequency derived alleles. We then talked about some of the effects of either sweeps or background selection on the entire genome. That's to kind of set up. Then we've got 6 lectures talking about actually testing for sweeps and soft sweeps and stuff. So before getting into that, let me give one last chance for any more questions or clarifications.